Welcome back, everyone. Ready for another deep dive?
Always excited to explore these intricate cellular mechanisms.
Today, it's all about the phosphoinositide cascade, a cellular chain reaction, really, and it plays a huge role in how our cells function. We've had a ton of requests to break this one down.
It's a fascinating system, really showcases how cells can amplify signals, like translating a message from outside the cell into action inside.
Exactly. And we're picking up from our last deep dive on G protein-coupled receptors, or GPCRs. They're like antennas on our cells, picking up signals from all over.
Hormones, neurotransmitters, even light. All sorts of signals can activate these receptors. And this phosphoinositide cascade we're talking about is one of the key ways those signals get turned into, well, cellular action.
Okay, so let's trace the steps. It all starts with a signal binding to a GPCR, right?
Right, and that activates a specific G protein inside the cell. It's the first step in the relay.
The G protein's like the first runner, passing the baton. So what happens next?
That activated G protein interacts with an enzyme called phospholipase C, or PLC.
PLC. I remember this from our notes. It's kind of the initiator of the whole cascade, wouldn't you say?
You got it. PLC's main job is to break down a certain phospholipid. It's called PIP2, and it's found in the cell membrane.
So PIP2 is just hanging out in the membrane, and then PLC comes along.
And chalks it up, yeah.
And what happens when PIP2 gets chopped up?
Well, that's how we get two super important second messengers, IP3 and DEG.
Ah, IP3 and DEG. The dynamic duo, as they're often called. But what do they actually do? What's their role?
Okay, so IP3 has a very specific target, the endoplasmic reticulum, or ER. Think of it as the cell's calcium storage facility.
So IP3 is like a messenger heading straight for the ER with a purpose.
Exactly. When IP3 gets to the ER, it binds to its receptor, and that triggers a release of calcium ions into the cytoplasm.
So it's like IP3 is saying, hey, ER, time to release the calcium. What about DAG? Where does it fit in?
DAG stays put in the cell membrane, but it's not just sitting around. It teams up with that newly released calcium.
Okay, so they work together.
Yeah, to activate another major player, protein kinase C, or PKC.
PKC. Now, this sounds like a powerful molecule.
Oh, it is. And the teamwork between DAG and calcium is crucial for activating it.
What's so special about PKC?
Well, PKC is a kinase, which means it can modify other proteins.
Modify. What does that mean in a cell?
It's like PKC is attaching little instructions to other proteins. These instructions change how those proteins behave. They might get switched on or off, and that affects a ton of cellular activities.
So PKC is kind of like a conductor, directing the activity of other proteins based on instructions from DAG and calcium.
A great analogy. PKC is involved in cell growth, differentiation, even cell death. It really does have its fingers in a lot of pies.
So to recap, we have this incredible chain reaction. A signal activates a GPCR, which turns on the G protein that leads to PLC activation, the breakdown of PIP2, calcium gets released, and finally PKC gets activated. It's like an intricate dance happening inside our cells.
A perfectly choreographed dance. And as with any good dance, if one step goes wrong, the whole routine can get messed up. That's where we start to see problems, even diseases.
Right. Our notes mentioned pathologies linked to disruptions in this pathway.
Exactly. If there's a mutation in a gene that codes for any of these proteins, or if the levels of these second messengers get out of whack.
Too much or too little activity can disrupt the balance.
And that can lead to disease, yeah. That's why understanding this pathway is so crucial. It's not just a theoretical concept. It has real-world implications for our health. And the amazing thing is researchers are actually figuring out ways to manipulate this pathway to develop new treatments.
That's what always gets me excited, taking that basic science and using it to help people, to improve lives.
Absolutely. Scientists are working on drugs right now, targeting specific components of this pathway, trying to correct imbalances, treat diseases at the source.
It's like learning the language of our cells so we can communicate with them, fix things when they break down.
Exactly. Like, there's been some really fascinating research on cancer, looking at how disrupting this pathway could be beneficial in treating certain types.
Oh, that's interesting. So not restoring balance,
Yeah.
but actually using the dysregulation to our advantage.
You got it. In some cancers, blocking certain parts of the phosphoinositide cascade can actually inhibit tumor growth. Pretty clever approach.
That's a perfect example of how really understanding these processes can lead to some unexpected strategies. What other research highlights were you impressed by?
Well, there's a ton of research going on right now looking at this pathway and its connection to Alzheimer's disease.
Yeah, I remember reading something about that. Something about calcium signaling being a key factor.
That's what they're trying to figure out. There's evidence that disruptions in this pathway, particularly those affecting calcium signaling, could be contributing to the neuronal damage we see in Alzheimer's.
So even though we're talking about this microscopic cascade, its impact can be seen at the level of the brain, contributing to these serious neurodegenerative diseases.
Exactly. And that's what I find so fascinating about cell biology. These tiny molecular events can have huge consequences for the whole body, the whole organism.
It really makes you appreciate how complex everything is, how connected our bodies are. Speaking of connections, one of the articles we looked at mentioned this pathway and the immune system.
Yeah, the phosphoinositide cascade is crucial for regulating immune responses. Immune cells, like any other cell, rely on these signaling pathways to work properly.
So when we get sick, this cascade is part of that whole network that helps us fight off infection.
Exactly. Activating immune cells, triggering the release of inflammatory mediators, coordinating the whole immune response. It's a complex dance, but this cascade is definitely part of it.
Amazing how this one pathway can have so many different effects, depending on the cell type and the situation.
It shows how fundamental it is. It's like a versatile tool, and cells can use it in different ways depending on what needs to be done.
It's easy to get lost in the details, but it's important to zoom out sometimes, look at the big picture. What's the biggest takeaway for you with all of this?
For me, it's realizing that the phosphoinositide cascade isn't just a simple chain of events. It's a dynamic network. It's interconnected with so many other pathways, and its influence goes way beyond just calcium release.
Right. It's like a central hub, integrating and amplifying messages from all these different sources to control all sorts of activities.
A great way to put it. And this understanding, it has huge implications for how we approach health and disease. Even small disruptions in this pathway can have cascading effects throughout the whole cellular network, contributing to all sorts of pathologies.
It really does make you realize how much our health depends on this delicate balance, these intricate processes happening inside us all the time.
It really does. And as we learn more about these mechanisms, we're gaining insights that could transform our understanding of ourselves and the world around us.
It's like we've zoomed in on this tiny microscopic world and discovered a whole universe of complexity and elegance. Pretty humbling, really.
It is. And the more we learn, the more we realize how much we don't know. There's still so many unanswered questions about this pathway, how it's regulated, its interactions, its role in specific diseases.
It's a good reminder that science is a journey, not a destination. And this deep dive, it's just a glimpse of how much there's still left to explore.
But that's what makes it so exciting. Every discovery opens up new avenues for research, new possibilities for understanding, new ways to manipulate these mechanisms.
So for our listeners who've been following along with us on this journey, what's the one thing you'd like them to take away from this?
I'd say it's an appreciation for the power of these tiny events. The phosphoinositide cascade is just one example, but there are countless signaling pathways operating within our cells. It's a symphony of life, really.
And by understanding these fundamental mechanisms, we can gain insights into everything, from normal physiology to the development of diseases.
It allows us to ask new questions, to come up with new strategies for treatment, and to just marvel at the beauty of the world inside ourselves.
What a perfect note to end on. We've journeyed through the complexities of this cascade, from the activation of those GPCRs to the release of calcium and the activation of PKC.
We've explored its role in all sorts of cellular processes, its implications in diseases, and the research that's helping us unravel its mysteries.
But this is just the beginning, as we've said. Science is an ongoing journey, and we encourage you to continue exploring this fascinating world of cellular signaling.
Keep asking questions, keep searching for answers, and never lose that sense of wonder.
Until next time, keep diving deep. You know, it's really wild when you think about it. Something as tiny as a calcium ion can have such a huge impact on so many different processes in our bodies.
Right. It's like a tiny little thing having this massive ripple effect. And here's something else that's super interesting. Research is showing that the phosphoinositide cascade isn't just about responding to signals from outside the cell. It actually might play a role in shaping those signals themselves.
Hold on, you're saying this pathway might be part of a feedback loop, so it doesn't just respond to signals, but helps to modify them too.
Exactly. It's like a two-way conversation between the cell and its environment, always adapting, making fine-tuned adjustments to those signals to keep everything in balance.
Wow, that's incredible. So it's even more dynamic and complex than we thought. It's not just a one-way street, but this multi-layered system with all these intricate feedback mechanisms.
You got it.
Yeah.
And figuring out how those feedback loops work, that's essential if we want to understand how cells maintain that delicate balance, that homeostasis, and of course how things go wrong when that balance gets disrupted and leads to disease.
It's like we've peeled back a layer, and there's a whole other level of complexity underneath. Makes you wonder what else we'll discover about this pathway.
That's the amazing thing about science. There's always more to learn. And every new discovery brings up new questions, new avenues for research.
And hopefully, new treatments, right? We've talked about some of the exciting possibilities, but I'm sure there's so much more to come.
Oh, absolutely. As we unravel the complexities of this cascade, I have no doubt that we'll find even more targets for drug development. That means more precise and effective therapies for all kinds of conditions.
It feels like we're entering a whole new era of medicine, where we can actually target diseases at the molecular level because we're getting a deeper understanding of those underlying mechanisms.
It really is an incredible time to be working in this field. The possibilities are pretty much endless.
Well, I think it's time for us to wrap up this deep dive. We've covered a lot of ground today, from the basics of the phosphoinositide cascade to its role in health and disease.
We've talked about how it amplifies signals, releases calcium, activates PKC, and how it's all orchestrated to create this amazing array of cellular events.
We've also explored what happens when things go wrong, when that regulation breaks down, and of course the incredible research that's paving the way for those new treatments we talked about.
But what we really hope is that we've sparked your curiosity, that you'll want to keep exploring this incredible world within our cells.
So what are you thinking about after this deep dive? What questions are still swirling around in your head? What else do you want to learn about this pathway?
The thing about a deep dive is, it's really just the beginning. It's a jumping off point for you to do your own exploring, to dig even deeper into those mysteries that are still out there.
We definitely encourage you to take what you've learned today and keep going on your own journey of discovery. There's so much to learn about this vast and intricate world of cellular signaling.
Keep asking questions, keep searching for answers, and most importantly, never lose your sense of wonder.
Until next time, keep diving deep.